stem cells and we use them from an old donor, they make many more myeloid cells than a young donor. So here we have mostly, I think, 70% myeloid cells compared to about 14% from a younger donor. So there's a difference in the age of the stem cell, but there's also a factor of how old is the niche that you're going into. So if we take stem cells and put them into an adult, whether the young or old stem cells, you'll get different levels of engraftment. Older stem cells engraft less well, young cells engraft at a higher level. However, if we put those in a younger animal with a younger niche, the environment, basically the transplant efficiency is about the same, indicating that the niche itself is very important for supporting both engraftment and hematopoiesis. And so potentially, can we reverse the aging process? If we change the niche and put an old stem cell, potentially we have ways to improve aspects of longevity and health by improving, again, the environment of the stem cell per se. So I think there is still hope for older stem cells, both by enhancing the environment and by different factors that we know can enhance hematopoiesis. So I have a few minutes left, and I'll switch gears a little bit. I'll only acknowledge for the first part, this work was done in the lab of my mentor, Weissman. A lot of the work on the ACK2 antibody was led by Mishka Cheshewich, who's a very talented medical student. The work on the stem cell niche I did together with Charles Chan and a crew of others in the lab, and that work is ongoing now to try and translate that to the human system and look at how we can manipulate both of those systems, both the antibody-based approach and the stem cell niche using anti-human antibodies and using human-derived cells to create human niches. So, last part of my talk, since about 5 minutes, is an aspect of regenerative medicine that relates again back to transplant and the niche and how do we get stem cells out. So, when we do a transplant in the lab, we get usually about 1 liter of bone marrow from a bone marrow donor. This is only about 1/2 liter in this picture. That's what it looks like. And how do we obtain that bone marrow from the donor niche per se? Well, we take a large trocar needle and we basically place it directly into the marrow environment. Usually do about 100 to 200 separate pokes into the bone marrow environment, and each time you enter, you can pull out about 5 to 10 mLs of bone marrow. That bone marrow is highly diluted with peripheral blood, by the way, because essentially while we get 1 liter of volume from that patient, there's not really even 1 liter of volume in the donor. So essentially, the donor by the end of the transplant procedure, who's usually a healthy volunteer or sibling, has a backside that looks like this. And as a transplant doctor, your arm is sore. You're doing this for about an hour in the operating room. And when I was doing my fellowship at Stanford, I thought there might be a better way to do this in a less invasive outpatient approach. Instead of doing hundreds of holes, maybe we could do just one and do this in a less invasive approach. And we developed a device called the marrow miner, which looks big and scary here, but I'll show you how it works. Essentially, it has a flexible catheter that enables you to enter into the marrow cavity, and is summarized in this short movie. And this enables us to get cells out of the niche, not only blood stem cells, but mesenchymal stem cells as well. Essentially, we can enter just once under local anesthesia. We have a flexible catheter with a rotating tip that can move through that crunchy part of the marrow, and this is faster than in real time, but lets us aspirate or suck out the bone marrow from the patient relatively quickly from a single hole under local anesthesia as an outpatient instead of going to the operating room. Enables us to very rapidly collect a bone marrow donation in a much faster, easier, less invasive way. So we've now taken this through clinical development, and the bottom line is it not only enables us to move through the marrow cavity more quickly, but when we compare this to the normal needle, if we compare a standard trocar, we'll get, if we look at the stem cell activity in a standard aspirate, we can look at the colony-forming unit assay activity. When we compare that with the marrow miner on the other side, we get about 10 times more stem cells out per unit volume than the standard approach. And when we took this into humans, this is actually a live patient using fluoroscopy. You can see the device following around the curve of the iliac crest. In one patient, in about 10 minutes here, we could collect about 500 mils of bone marrow. And not only did we have bone marrow in 10 minutes with one entry site, we had, overall, compared to the standard approach, about 5 times more hematopoietic stem cells and about 8 times more mesenchymal stem cells in the standard approach. And now we're in the process of through a spin-out company called SENS4, applying this to other fields, including orthopedics, where adult bone marrow-derived cells are used, potentially treating vascular disease. There's a lot of work been done in treating cardiovascular disease with marrow-derived cells, and the hope is, using this sort of system to harvest autologous bone marrow, we can hopefully speed up and enable the field of regenerative medicine when it's using patients' own cells. And the device will hopefully be on the market by early next year. So hopefully today I've covered 3 aspects: the bone marrow niche, how we can better manipulate that with antibodies, how we can understand it, and here's an example of a way to harvest cells from the stem cell niche. And I'll mention 2 things. This may, number one, enable you to bank your own bone marrow when you're younger and healthier to potentially use if you need it when you're older or have a malignancy or other indication. And also may be an approach to deliver stem cells back to the bone marrow, or to deliver a new niche in a patient that has aplastic anemia or some other disease of the niche itself, in myelodysplastic syndrome or others, could be used as a way to deliver cells to the marrow environment as well. So I'll stop there and take questions. Thanks.
Thank you very much.